I'm Dr. Sarah Comstock, and we will be discussing Chapter 24, the digestive system. And even though we covered this in lecture in person, I'm starting from scratch or from the beginning because I want you to be able to go back and play this all just in case you want to do some reviewing on your own. So as a reminder, most of the digestive system anatomy that you see here was covered in lab. And you might have a little bit of the content quizzed on, but in general, most of that is going to be covered in your lab section of this course. So for the first portion, we're going to be talking about sections 24.1 through 24.3. There are two groups of organs that compose the digestive system. The first is the gastrointestinal tract, or also called the GI tract, and basically this is a long tube, and it goes from the mouth all the way to the anus, and it's going to contain mouth, most of the pharynx, the esophagus, stomach, small intestine, and large intestine, and ends with the anus. Then the other organs that you have that are not necessarily part of the GI tract are your accessory digestive organs, and these include teeth, tongue, salivary glands, liver, gallbladder, and pancreas. And these contribute to digestion, each in their own way, that we're going to learn about later on. The GI tract has 4 different layers, and so we're going to go through them starting from the innermost layer, which is called the mucosa. And the mucosa is basically a thin layer of smooth muscle that actually makes folds in the stomach and small intestine, and the purpose of those folds is to increase surface area. Next, we have the submucosa, and the submucosa is made up of connective tissue, and this binds the mucosa to the muscularis. And it's full of different blood vessels, and it actually has a plexus of nerves, which is basically just an interconnection of nerve fibers. This plexus is called the submucosal plexus. The muscularis is the third layer, and this is composed of muscle, as you can tell from its name. In the upper portion of this long GI tract, you'll have voluntary muscle, and that composes the muscle that's in the mouth, the pharynx, and upper 2/3 of the esophagus. In addition, the anal sphincter is also a voluntary muscle. The rest of the muscle in the muscularis layer is involuntary, and this is what makes the involuntary contractions that are helpful in peristalsis throughout the GI tract. You have 2 layers of circular and longitudinal muscle, or you have a layer of circular and a layer of longitudinal muscle, so it makes up 2 layers of muscle in this region. In addition, the stomach has a third oblique layer. And then there's another plexus there called the myenteric plexus that's between the muscle layers. Finally, the serosa is what surrounds everything. And this is basically connective tissue and epithelium. However, it's not present in the esophagus. Moving on to neural innervation, the GI tract is controlled by 2 sets of nerves. An intrinsic set of nerves that we call the enteric nervous system, so that's within the GI tract, and then an extrinsic set of nerves that are part of the autonomic nervous system. So we've talked about the sympathetic and parasympathetic nervous system previously, so we're going to see how those are regulating our GI tract. The enteric nervous system, we talked about these 2 plexuses earlier, the submucosal plexus that's in the submucosal layer, and the myenteric plexus that is in the muscularis layer. And if you're wondering what this term plexus means, it's basically just a branching or interconnected branching bundle of nerve fibers. So that's what we have there, those 2. And then let's look at autonomic nervous system. We have 2 branches of the autonomic nervous system, the sympathetic and the parasympathetic. And we know that the sympathetic is the fight or flight, but the one that's activated in digestion, parasympathetic, is our rest and digest nervous system. So our parasympathetic is going to regulate our digestive organs as well. Doing a quick review of the nervous system, recall you have a central nervous system and peripheral nervous system. And then the peripheral nervous system is subdivided into the somatic nervous system that's going to regulate skeletal muscle, the autonomic nervous system that's going to regulate smooth muscle, cardiac muscle, and glands, and finally what we just talked about, the enteric nervous system that is going to regulate smooth muscle and glands, but these are part of the GI tract. So now we understand how this all fits together with our nervous system. We call the enteric nervous system the brain of the gut, and this is basically because it consists of sensory neurons, interneurons, and motor neurons that are going to sense things in the GI tract. Interneurons are going to do some of the integration, and motor neurons are going to be the output of the nervous system here. So recall you have sensory neurons that sense and bring input in, interneurons integrate, and motor neurons are the output neurons. And these neurons are going to extend all the way from the esophagus to the anus. And again, reminding you, we have two plexuses. The myenteric plexus is what's going to regulate motility. And again, it's in the muscularis layer, so motility is controlled by the muscles, so that makes sense. Submucosal plexus is going to be what controls secretions. So secretions are going to be secreted from the submucosal layer, so that also makes sense why you would have that in that location. Let's use this image here to help you understand what I mean by sensory, inter, and motor neurons. So I'm going to highlight the sensory neurons. They're going to sense what's occurring in the mucosal epithelium here. And they're going to send action potentials that are going to integrate in a couple different areas. Some are going to integrate in the autonomic nervous system and the central nervous system, or they can integrate in the submucosal plexus or in the myenteric plexus. We have interneurons that are going to do the communication between your sensory neurons and your motor neurons. And then your motor neurons are going to give you the output. So here, if we're looking at myenteric plexus, it's mostly going to control the GI tract motility, and that word means movement. And that's particularly how frequent and how strong the contraction of the muscle layer is going to be. If we look at the submucosal plexus, again, let me remind you that this one controls secretion of organs of the GI tract. So here's our submucosal plexus, and if we're going to control secretions, we're going to actually have to have that occur via motor neurons. Sensory neurons are going to sense the environment in the mucosal epithelium, but the action potentials that are going to lead to those secretions are going to occur via those motor neurons. What do you need sensory neurons for if the action potentials that lead to secretions are coming from motor neurons? Well, again, sensory neurons are going to sense the local environment. So we're going to have chemoreceptors at the ends of the sensory neurons. They're going to be activated by chemicals in food, that these are going to then tell the submucosal plexus neurons what secretions need to occur. And then you're going to have stretch receptors that are going to be activated when food stretches the walls of the GI tract organs. And then this is going to tell the myenteric plexus neurons to send action potentials via motor neurons for contractions of muscles. We talked about how the enteric nervous system is the brain of the gut, and it can work independently, but it can be regulated by neurons from the autonomic nervous system. As a reminder, the parasympathetic nervous system is what we call the rest and digest system. So it actually stimulates increases in secretion and activity by stimulating the enteric nervous system. The sympathetic nervous system actually decreases secretion and activity, and this is our fight or flight response system. And we don't really need to be digesting food when we're running away from a bear, right? When we're in a fight or flight situation, we are not in need of digesting food. So this sympathetic activation is going to decrease or inhibit the enteric nervous system. So we're finishing up this section, and I'm going to ask you one question to see if you remember what we talked about earlier. All the following are considered organs of the gastrointestinal tract except for, and I actually am pointing at it here. The answer is gallbladder. The gallbladder is considered an accessory organ, but it's not a GI tract organ. Moving on to sections 24.4 through 8. This is actually going to be a quick overview. A lot of the rest of the stuff is covered in your reading guide, but we're just going to do this pretty quickly. I want you to understand that there are 6 basic processes involved in digestion. Ingestion, to begin with, is basically just putting the food in the mouth and then swallowing it. Second, you have secretion. Well, secretion is going to be glands, digestive glands, attached to your GI tract or within your GI tract that are going to secrete digestive enzymes. The motility is mixing or propulsion of foods and the secretions together. Digestion itself is actually taking those larger food particles and using digestive enzymes is breaking them down into smaller food molecules. Absorption is going to be bringing the smaller food molecules across the wall of the GI tract into blood or lymphatic vessels. And finally, defecation, which is going to be eliminating the waste and the things that aren't digested via the anus. So digestion is going to begin in the mouth. We're going to start with mechanical digestion, which just occurs via chewing. If you're chewing or masticating, you're going to be breaking up those food items into smaller particles. Then you have chemical digestion. Your salivary glands secrete salivary amylase, and this is going to act on starches and glycogen, and it's actually going to continue to act until it's the enzyme itself is inactivated by stomach acid. The lingual glands of the tongue secrete an enzyme called lingual lipase, and this acts to start breaking down triglycerides, but it actually doesn't become activated until it hits the stomach. So you secrete it in the mouth, but you don't actually have active lingual lipase until it hits the stomach. So let me ask you this question. Deglutition. What does that mean? The answer is D, the act of swallowing. And you should have read about this in your reading prior to coming to class, and you probably already talked about it in lab. So the act of swallowing occurs. It's a voluntary activity because you actually use the muscle in your mouth voluntarily to basically push a bolus of food down your esophagus. And what's important to note here in this figure is that the epiglottis is going to close off the opening of the larynx when you swallow. So let me highlight here. This is the larynx and this is the epiglottis. And you'll note right here that that opening right there is closed off. And that's pretty important because what's the larynx? Well, it's the opening to the windpipe or the trachea. And you do not want to get food in your windpipe or your trachea. That's what's going to cause coughing, because if you did get it in there, then it might go into your lungs. So, deglutition. Once you swallow that bolus, it enters the esophagus, and peristalsis, which is muscular contraction of muscles or contraction of muscles, is going to push that bolus of food down until it hits the stomach. Let's define peristalsis. It's a series of contraction and relaxation of the circular and longitudinal muscles of the muscularis layer. So basically, those muscles are going to crimp together and then slide down repeatedly over and over and over again to move the bolus of food down the esophagus into the stomach. And you should note that peristalsis occurs in a lot of other tubular organs, including the GI tract that we'll keep talking about, but also things like ureters, bile ducts, and uterine or fallopian tubes. This happens a lot in our tubular organs. So that's all I'm really discussing for sections 4 through 8. The remainder of the objectives are going to be covered in your reading guide and also the lab. Moving on to sections 24.9 through 11, we're going to go more in depth here on the stomach, the pancreas, the liver, and the gallbladder. So the stomach is going to serve as a chamber for holding and mixing this thing called chyme. What's chyme? Well, chyme is the mixture of food plus digestive enzymes that are secreted into the stomach. So I'm going to ask you a question. Hydrochloric acid and intrinsic factor are secreted by which cell type in the stomach? The answer is parietal cells. All right, so you have multiple different cell types in the stomach. And these cell types are going to exist within what's called these gastric pits. The gastric pits are these indentations or invaginations of the lining of the stomach. And then at the very base of that, you have gastric glands. And there's certain cell types in there that secrete enzymes that are important for digestion. So let's start with the cell types that are towards the surface of the gastric pits, and these secrete mucus. So you have two types. You have mucus or surface mucus cells and mucus neck cells, right? And so they're just going deeper and deeper into these gastric pits. As you get deeper, you're now calling them gastric glands, and we have parietal cells, and if you selected A, you were correct on the previous question. Parietal cells secrete hydrochloric acid and also another enzyme called intrinsic factor. Chief cells secrete pepsinogen and gastric lipase. And then finally, G cells secrete the hormone called gastrin. So all of these secretions together, the secretion of mucus, parietal, and chief cells form gastric juice. And the mixing of the gastric juice with the food is what we end up calling chyme. So why do you think you need to secrete mucus? I think we've asked this in class before, but if you think about it, hydrochloric acid that's secreted from parietal cells can be pretty caustic to the stomach. So having that mucus secreted is going to line your stomach and help prevent hydrochloric acid from actually digesting your stomach tissue and using it instead to activate or help with digestion of other tissues. We talked about mechanical and chemical digestion in the mouth, and we're going to have the same thing occurring in the stomach. Mechanical digestion is what produces that thing called chyme, which is just the soupy liquid of all the enzymes and the food all together. And it occurs because of the churning movement that happens in the stomach. Chemical digestion begins with an enzyme called amylase, and here we're talking about salivary amylase. And salivary amylase, where do we secrete it from? We're secreted from salivary glands, and it continues to work in the fundus of the stomach. So where's the fundus? It's actually that upper curvature, and it's going to continue to work until it hits the more acidic gastric juice, and then salivary amylase becomes inactivated. We talked about digestive enzymes in the mouth. The other one that we talked about was lingual lipase, and this one actually doesn't become activated until it meets with the hydrochloric acid that is present in the stomach. And once it becomes activated, it starts to digest triglycerides into fatty acids and glycerol. So if you look here at this triglyceride, this is considered a triglyceride because it has this glycerol backbone and 3 fatty acids right here. So if we talk about it being a diglyceride, it means it's going to have lost one of the fatty acid chains, and it has 2 fatty acid chains attached to a glycerol molecule. The stomach secretes hydrochloric acid from parietal cells. In reality, it actually secretes hydrogen ion and chloride ion, which combine to form hydrochloric acid. And why do we need hydrochloric acid? Well, hydrochloric acid is very acidic, so it makes the pH of the stomach juice about 1.5 to 2. And that is important because it's going to kill microbes and it's actually going to denature proteins. Hydrochloric acid also stimulates the secretion of some hormones that promote the flow of bile and pancreatic juice. And those are going to be important for digestion later in the small intestine. Finally, it converts the inactive form, pepsinogen, into the active enzyme, pepsin. So that's going to be important as well because we don't want the active form in our cells, otherwise we'll start digesting our cells. So pepsinogen is secreted, and that is activated by hydrochloric acid and converted into pepsin. There are a variety of different factors that can increase hydrochloric acid secretion from parietal cells. One I want to point out to you is acetylcholine from parasympathetic stimulation. So parasympathetic stimulation, we understand, is our rest and digest system. And what we see here is that when that stimulation is increased, it's going to increase hydrochloric acid secretion, which is important for digestion. Gastrin and histamine are also going to be important for increasing hydrochloric acid. So both of those are also going to increase digestion. Let's go back and talk about pepsinogen again for a minute. So chief cells secrete pepsinogen, but hydrochloric acid secreted by parietal cells are what's going to take this inactive enzyme called pepsinogen and convert it into the active form called pepsin. And pepsin is important because it digests proteins. So this is one of the enzymes secreted from your cells of your stomach that help with protein digestion. We also talked about lipase before with lingual lipase. Now we have another enzyme called gastric lipase, and this one goes and splits triglycerides into fatty acids and monoglycerides. There's only a little bit of nutrient absorption that occurs in the stomach, some water ions, short chain fatty acids, some drugs like aspirin, and alcohol can be absorbed in the stomach. But in reality, most of the nutrient absorption is going to occur in the intestines. The stomach is going to empty into the first part of the small intestine called the duodenum. And we also have right here an accessory digestive organ called the pancreas. And pancreatic juice is secreted from the pancreatic duct into the small intestine. If you were going to look at the histology of the pancreas, as in looking at the cells of the pancreas under the microscope, you would notice that 99% of the cells are exocrine cells. They secrete pancreatic juice, which is a mixture of fluids and digestive enzymes, into the pancreatic duct. Only about 1% of the cells are endocrine cells, and these are islets of Langerhans, and we talked about them before when we talked about the endocrine system. The cells within the islets secrete insulin, glucagon, somatostatin, and pancreatic polypeptide. But the big focus for us, for the pancreas, are those exocrine cells. So we're going to look at this a little bit closer. You will see that we have the pancreatic duct. I'm going to highlight it right here, right? And so this is this long thing called the pancreatic duct. And then we have an accessory duct as well, right here. Well, if you continue down on the pancreatic duct and zoom in, you'll notice that it merges with the common bile duct. And I'm going to highlight the common bile duct right here, which in this image is green. Together, when they merge, the new duct right here is called the hepatopancreatic ampulla, or the ampulla of Vater, right? And together they are going to dump into the duodenum. And what they're going to do is they're going to secrete pancreatic enzymes from pancreatic juice or bile directly into the duodenum, which is going to enable you to digest either proteins or fats. So let me ask you this question. Pancreatic juice does all of the following except. Which one does pancreatic juice not do? And your answer is provide glucagon. Yes, glucagon is secreted from the pancreas, but it's secreted from endocrine cells, not exocrine cells. Endocrine cells secrete glucagon, and if you recall, endocrine means it's going to secrete directly into the blood. You're not going to secrete into the lumen of your GI tract, but instead into the blood. So that's why that one's not true. But the other things are true. So pancreatic juice does buffer gastric juice. It does provide protein digesting enzymes in the inactive form. It provides starch digesting enzymes, and it also stops the action of pepsin. Okay, so what is pancreatic juice composed of? Well, in reality, it's mostly water, but then there's a few things dissolved in it. Sodium bicarbonate is actually going to neutralize the acidic stomach chyme, and this is what's going to help inactivate pepsin. And then there's a variety of different enzymes. One of them, the first one, is an amylase, so it's pancreatic amylase. So remember, we've seen amylase before. This one is going to help with digestion of carbohydrates. You have a variety of proteolytic enzymes. And when I say proteolytic, that means they secrete proteins. And they actually secrete these enzymes in their precursor forms, just kind of like we learned that pepsinogen was secreted. So trypsin is actually secreted as trypsinogen. Chymotrypsin is secreted as chymotrypsinogen. Carboxypeptidase as procarboxypeptidase. And elastase as proelastase. And these are all going to help with protein digestion. You have pancreatic lipase. We learned that lipases help with digestion of lipids or fats. And finally, we are able to digest RNA and DNA. And the enzymes that do this are ribonuclease, which digests RNA, and deoxyribonuclease, which digests DNA. Moving on from the pancreas to two more accessory digestive organs are the liver and the gallbladder. The liver is composed mostly of cells called hepatocytes, and these hepatocytes make something called bile, which is just an excretory product and digestive secretion that's going to help with emulsification of fats. Well, what do I mean by emulsification? Basically, it's going to take large fat globules and turn them into a lot smaller little fat droplets, which enables enzymes like lipases to come along and digest them more easily. Now, the liver makes the bile, but the gallbladder is what stores the bile until it's needed. So you'll notice you have two ducts coming from both sides of the liver, the right and left hepatic duct. And bile is going to come down from there and they're going to join into the common hepatic duct. And together with the cystic duct, which comes from the gallbladder, this is going to form into the common bile duct. And recall that this joins up with the pancreatic duct to form the ampulla of Vater. So all of these, the bile that's secreted from the liver gets concentrated in the gallbladder and then dumped along with pancreatic juice into the duodenum via that common duct. If you were looking for the gallbladder, you would actually find it located on the posterior surface of the liver, just on the basal and kind of what I'd say underneath the surface of the liver if someone was laying down. And I talked about this a little bit ago, but basically the function of the gallbladder is to store bile. It doesn't actually make the bile. The bile is produced by the liver, but it's stored in the gallbladder until it's needed in the small intestine. And it absorbs water and ions in order to concentrate the bile even more than when it's produced from the liver. And finally, contractions in smooth muscle are what around the gallbladder are what is going to eject the contents of the gallbladder into, or this bile, into the cystic duct, which then joins up and dumps its contents into the duodenum. The liver is composed of hepatocytes. That's the major cell that you're going to find in the liver. But it also has these green things called bile canaliculi, right? And hepatic sinusoids. So basically what you see with the hepatic sinusoids is that they're these highly permeable capillaries that allow blood to flow from the portal veins, or a branch of the portal vein, into the central vein. Looking at this diagram, tell me what you see indicated by C. These are actually the hepatic sinusoids, and these are going to run from the hepatic veins and drain directly into the central vein, which you see right in the middle, which is a C. So these are really permeable capillaries that drain from a branch of the hepatic portal vein into that central vein. And you're going to see this all over if you're doing histology of the. Let's go back to bile. Your hepatocytes secrete almost a liter of bile every day. And it's made up of mostly water, but it also contains biosalts, cholesterol, lecithin, biopigments, and several different ions. So it's partially an excretory product, but it's also partially a digestive secretion. And then let me explain how. So the principal pigment, I said there's several bile pigments. The principal pigment that is present in bile is bilirubin. And this is actually derived from the heme of red blood cells after they're phagocytized. So basically you're breaking down the heme and releasing this bilirubin into bile, and this is what's going to give it its greenish color. But also, bile is important in emulsification of fats, and we talked about this a little earlier, but basically bile salts are going to enable the breakdown of large fat globules into smaller fat droplets. And that's really important because it provides more surface area for enzymes, such as the different lipases we learned about, to work and digest fats. The liver does have other functions. For example, with carbohydrate metabolism, it's going to convert glucose to glycogen in order to store high amounts of sugar when blood glucose levels are high. And it also breaks down glycogen into glucose. So when blood glucose levels drop, then it's going to break down glycogen to produce more glucose to be released into the bloodstream. Now we're going to cover sections 2412 through 14, and this is going to be the small and large intestine and phases of digestion. The small intestine is where the majority of digestion and absorption occur, and it has three regions. The first region is called the duodenum, or you can also say duodenum. Both are correct. Then jejunum, and finally ileum. And the ileum is what it connects to the large intestine. The small intestine has four main functions that we'll discuss. The first is a mixing process, and it's actually occurring via segmentations, and they mix chyme with digestive enzymes, and this brings the food in contact with mucosa so that it can absorb the nutrients from the food. Segmentations are localized mixing contractions, and these occur in different portions of the intestine when they're distended by food. The second process is peristalsis, and peristalsis is just going to propel the chyme through the small intestine. We talked about this earlier when we talked about peristalsis in the esophagus, and this is going to occur all throughout the GI tract. Third is digestion. So the small intestine digests, or actually completes digestion of carbohydrates, proteins, and lipids. And it begins and completes digestion of nucleic acids. And finally, nutrient absorption. About 90%, or the majority of nutrients and water that pass through the digestive system are absorbed in the small intestine. To understand the function of the small intestine, we first need to be familiar with the structure. So there are three structures in the small intestine that are necessary to increase the surface area. The first is circular folds, also called plicae circularis. The second would be villi, and the third is microvilli. So starting with circular folds, these are ridges, and they cause chyme to spiral as it moves down the small intestine. And these are going to increase the surface area so that more digestion and absorption can occur in the small intestine. Then the next thing that increases surface area are villi. And these are on top of those circular folds. You're going to have finger-like projections of the mucosa. And they contain arterioles, venules, capillaries, and lacteals. Lacteals are just basically lymphatic capillaries. And finally, you have microvilli. Microvilli are projections from cells that line the villi, and they're really, really hard to see. What you're seeing here in the first image is villi, right here. And it's on top of those villi that if you zoomed in really, really high with the scanning electron microscope, you'll see microvilli. And so what we call them is brush border because it's just this really wavy border that's really hard to see under a microscope. And those cells in the brush border secrete enzymes that we refer to as brush border enzymes. Your intestines secrete about 1 liter of intestinal juice daily, and this contains water and mucus, and it's slightly alkaline. And just having this liquid medium is going to help with absorption. The other thing that your intestine secretes are brush border enzymes. Brush border enzymes are secreted by cells that have the microvilli that line the intestine. Brush border enzymes are going to break down food products, mostly carbohydrates, protein, and nucleotides. And this enables some enzymatic digestion to occur on the surface of those microvilli rather than just in the lumen from other digestive enzymes. Now that we've talked about the structure of the small intestine, I want to actually talk about the process of digestion that occurs in the small intestine. And recall, we divide digestion into chemical and mechanical. So beginning with mechanical, we have the myenteric plexus, which is located in the muscularis layer. We talked about this earlier, and it's just a bundle of nerve fibers, interconnecting nerve fibers. And this regulates mechanical digestion because it regulates segmentations. Segmentations, again, let me remind you, are localized mixing contractions that we talked about earlier, and they mix chyme and also bring it in contact with mucosa for absorption. And they do not actually push the intestinal contents down the tract of the intestine. It's peristalsis that does this. Chemical digestion also occurs in the small intestine, and remember that about 90% of your nutrients are absorbed in the small intestine, so we really need to break them down. Carbohydrates are broken down via this enzyme we talked about earlier, pancreatic amylase, and also via brush border enzymes, including alpha dextrinase, sucrase, lactase, and maltase. And they all break down larger long chain sucrose or sugar molecules or starch molecules into smaller single monomers called monosaccharides. So the intestinal lining can actually absorb those monosaccharides. Proteins are digested via these enzymes: trypsin, chymotrypsin, carboxypeptidase, and elastase. And we talked about them earlier when we talked about the enzymes that are secreted from the pancreas. In addition, the brush border secretes two other protein digesting enzymes called aminopeptidase and dipeptidase. And these break down your proteins into smaller molecules that can later be absorbed. Lipids are broken down via this enzyme called pancreatic lipase, and it's really the most important enzyme in triglyceride digestion. But bile salts are critical as well because they emulsify. And we talked about this earlier. Recall that emulsification is just going to break down those larger fat globules into smaller fat droplets, and this is going to increase surface area so that enzymes like pancreatic lipase can come in and digest. Finally, nucleic acids are broken down via these two enzymes, ribonuclease and deoxyribonuclease, secreted from the pancreas, and nucleosidases and phosphatases secreted by brush border cells. So these are all the different enzymes that are secreted and are working in the small intestine. So what do you need to know for the test? For the test, I'm going to want you to know the enzyme type and what it digests and what cell type secretes those enzymes. So if that makes sense to you, I want you to know the names of the enzymes, what they digest, and where they're secreted from. Table 24.5 in your book will give you a nice summary of all these different enzymes. Chemical digestion is the breakdown of larger food particles or molecules into smaller molecules. But once digestion occurs, we then need to absorb those molecules into the body so that they can be used as a fuel source for other processes in the cell. Here we see that carbohydrates are broken down into smaller monosaccharides, either galactose, glucose, fructose, or others, that are then transported or absorbed into the cells. And these can occur a couple of different ways. Glucose and galactose are going to be transported via a secondary active transporter that uses sodium down its concentration gradient to pull in glucose and galactose. Fructose, on the other hand, just diffuses through the cell membrane. And then they're all able to diffuse into blood capillaries after that point. Proteins are broken down into individual amino acids or larger ones called dipeptides or tripeptides. And there's an active transport process that also brings those into the cells lining the intestine, and then they diffuse out of those cells into blood capillaries. Lipids are a little different. If it's a small short-chain fatty acid, it will go through the cell via simple diffusion and then out into blood capillaries. Larger, long-chain fatty acids or monoglycerides, and we'll talk about those in a second, are going to be able to diffuse in, but then they're transported via this thing called chylomicrons. And chylomicrons are just a group of lipids that are surrounded by a protein outer casing. And instead of diffusing straight into capillary blood, they actually go into lacteals of the villi that line the surface of the intestinal tract. And from there, they're going to be brought into the blood via a different manner. So let's look at this a little bit more closely. As you can see here, monosaccharides, amino acids, and small chain fatty acids or short chain fatty acids diffuse into the blood of the capillaries that are within the villi. And then they're going to be transported via the hepatic portal vein to the liver. And then the liver is going to have circulation that allows for transportation of those nutrients all throughout the body. On the other hand, the chylomicrons that we talked about earlier are not going to be dumped directly into the capillaries. Instead, they go via these lacteals that you can see here in green, and those join up with the thoracic duct, and then those dump back in to the blood via the subclavian vein. Let's address each nutrient separately. So, monosaccharides are going to be absorbed via either secondary active transport or facilitated diffusion. And then they're going to be delivered to the bloodstream again via facilitated diffusion. Any carbohydrate that's digested or broken down into a monosaccharide can be absorbed. However, there are some undigestible celluloses or fibers, and since they can't be broken down into absorbable monosaccharides, they're going to be left in the feces. And you can see right here this little circle that's surrounding one of those red monosaccharides. So this long thing is a glucose polymer, and it's all branched, but the red thing is what we call an individual monosaccharide. Amino acids, dipeptides, and tripeptides are also able to be absorbed into the blood. So if you look at this polypeptide chain on the right, one of those individual blue squares is an amino acid. A dipeptide or a tripeptide is 2 or 3 of those amino acids joined together via a peptide bond. And this takes active transport to bring them into those brush border cells, and then diffusion enables them to go into the bloodstream to be used as a nutrient source. Finally, we should address lipids. And again, lipids are a little bit different. As you see here on the left, this is a triglyceride, and what it has is 3 long-chain fatty acids attached together via this blue molecule, which is a glycerol molecule. So with those 3, we call it a triglyceride. On the right, you have short-chain fatty acids. And we call them short-chain fatty acids because you can see here, looking at the carbon molecules, they have 2, 3, or 4 carbon molecules. And when you compare them to the long-chain fatty acids on the left, they're shorter. So that's why we call them short-chain fatty acids. Lipases that we learned about earlier actually are able to break down those triglycerides into monosaccharides and fatty acids. So the gray thing we see here is a fatty acid molecule, and when it's attached to the glycerol molecule, it's called a monosaccharide. See the fatty acid molecule and the glycerol molecule. So like I said before, short-chain fatty acids go into the blood for transport via a simple diffusion process. They don't require a carrier. They're small enough and lipid soluble enough that they can go directly into the blood. On the other hand, long-chain fatty acids and monoglycerides are transported differently. These are very large and they're hydrophobic, so they're transformed into chylomicrons that you see there. And they're basically lipids coated with proteins. And then what they do is they enter the lacteals, where they eventually do get into the blood with a protein coat of chylomicrons, keeping them suspended and separate from the rest of the blood. We've talked about all the nutrients that your intestine absorbs, but we haven't talked about water yet. So you have about 9.3 liters of fluid every day that enter the small intestine. 2.3 liters of this is from ingesting liquids, but the rest comes from gastrointestinal secretions. So that's about 7 liters remaining. And the small intestine ends up absorbing 8.3 liters of water. And that leaves only about 1 more liter left, of which the large intestine absorbs 0.9 liters, leaving only 100 milliliters of water that's excreted in feces every day. So you can see that the small intestine also absorbs a great deal of water in addition to all the nutrients it absorbs. Let's look at the anatomy of the large intestine. The ileum joins up with the cecum at what is called the ileocecal sphincter or ileocecal valve. Attached to that cecum, you have an appendix, and then you have your ascending colon, transverse colon, descending colon, and sigmoid colon. And they call it sigmoid because it's S-shaped. From there you have the rectum and the anal canal. And you'll notice, fortunately, there's two sphincters. You have an internal anal sphincter and an external anal sphincter. Both mechanical and chemical digestion occur in the large intestine. So the gastroileal reflex is a reflex that occurs after a meal, and what it does is it forces a large amount of chyme into the cecum from the ileum through the ileocecal valve. That valve normally remains closed, but after a meal, there's a large propulsion of the chyme that goes into the cecum. Then there are a few other mechanical digestion processes. The first is haustral churning. So the haustra are the little pouches, or actually big pouches, that you see in the large intestine. And when distension reaches a certain point, what happens is the walls of the haustra contract and they squeeze the contents forward. So each one of those pouches squeezes separately. Then you have peristalsis, which is what we've talked about before. It's those propulsive contractions. But then you have a form of mass peristalsis, which is really just a strong peristaltic wave that begins at that transverse colon, and it just drives food quickly from the colon into the rectum, which again is going to initiate another response. And actually what happens is food in the stomach initiates this gastrocolic reflex, which makes you have to poop after a meal. So that's why people usually should poop about 3 to 4 times a day. Finally, we have chemical digestion in the large intestine, and this final stage of digestion is actually done through the bacteria that are present in the large intestine. These bacteria are going to convert proteins into amino acids, and they're going to break down those amino acids. They also ferment carbohydrates, which is what produces the gas that causes gas in the large intestine. And they also can produce some B vitamins and some vitamin K. Finally, you should know that mucus is secreted in the large intestine, but no enzymes. So this is why that final digestive process has to occur via the bacteria, not via enzymes secreted for the large intestine. And like I indicated earlier, about 0.9 liters of water is absorbed, along with some other ions and vitamins, is absorbed into the large intestine. So what this does is basically dehydrates the chyme, so it's no longer a liquid and it's a more solid stool. There are some structural differences between the large and small intestine that you should note. So when we look at the histology of the large intestine, we no longer see the circular folds or the villi. We do see a little bit of microvilli on absorptive cells, but in general you don't need the same structures that increase surface area because you're doing a lot less absorption in the large intestine than in the small intestine. You'll see that those little lines that are on the surface, they're called intestinal glands. So instead of the villi going up, the glands go down. And within the glands, you have goblet cells. And those goblet cells secrete mucus. Now, you have mucus secreting cells in the small intestine too, but as you go down the small intestine and continue to the large intestine, you have more and more mucus secreting cells. And as one of my professors of anatomy and physiology told me, the idea here is to grease the chute. Basically, you're secreting more and more mucus while you're absorbing water going down the intestine. So the feces, once they become concentrated, have a way of getting out. One major goal of the colon is to form feces. So the colon absorbs a lot of water, electrolytes, and vitamins to concentrate the feces. And feces are going to consist of a little bit of water, inorganic salts, epithelial cells that have been sloughed off, the bacteria, some of it that's present in your colon, some of the products of bacterial decomposition, and then any of the food portions that you ate that you're unable to digest. So let's talk about the defecation reflex. This is from a book called Everyone Poops. I thought you guys would enjoy that. It's a children's book. Maybe you can get it and read it if you'd like. So the reason you have a defecation reflex is because when feces enter the rectum and the rectum distends to a certain point, stretch receptors send nerve impulse to the spinal cord, and the region of the spinal cord that it stimulates is the sacral region. And then motor impulses travel back to the descending colon, the sigmoid colon, rectum, and anus, and what they do is they cause contraction of muscles, and it causes opening of the internal anal sphincter. And this is what pushes poop out. There are 3 overlapping phases in digestion: the cephalic phase, the gastric phase, and the intestinal phase. The cephalic phase of digestion occurs when sight, smells, thoughts, and taste of food stimulate the salivary glands to secrete saliva and the gastric glands of the stomach to secrete gastric juice. So have you ever walked into a restaurant and smelled food and your stomach started gurgling, or you thought of food, or you looked at food and you started salivating? This is caused by the cephalic phase. The gastric phase has both neural and hormonal regulation. So basically what happens is food causes stretch in the stomach, and this stimulates stretch receptors, and also chemoreceptors in the stomach are going to monitor pH. So let's look at this diagram to the right. If you see here, food enters the stomach, and what it does is it disrupts the controlled condition. It changes the pH of the gastric juice, and it distends or stretches the stomach wall. In response, certain receptors, which are chemoreceptors and stretch receptors, are going to detect this change, and they're going to send nerve impulses to the submucosal plexus. Remember, we talked about this plexus before, and that sends nerve impulses to the parasympathetic nervous system. And that causes regulation of effectors, and the 2 effectors we're seeing here are parietal cells that secrete hydrochloric acid, and the stomach has smooth muscle in its walls, and this causes contraction of that smooth muscle. So in response to this stimulation, there's an increase in acidity of the stomach chyme and a mixing of the stomach contents as well as emptying of the stomach. And what you see is that it returns to homeostasis via these responses. You also have hormonal responses in the gastric phase. The hormone gastrin stimulates gastric glands to secrete gastric juice, and then it also strengthens the contraction of the lower esophageal sphincter, and what that does is it prevents backflow or reflux of the food in the stomach, and it relaxes the pyloric sphincter, which allows for chyme to move from the stomach into the duodenum. The final phase of digestion is the intestinal phase, and this starts when food enters the small intestine. And all of this is going to be designed to slow the exit of chyme from the stomach. And the point of that is to prevent the duodenum from getting overloaded with chyme right after you eat. The intestinal phase has both hormonal and neural responses. There are two major hormones that are secreted in the intestinal phase, cholecystokinin and secretin. So cholecystokinin is secreted by these cells called CCK cells that are in the small intestine. And they sense the amino acids and fatty acids that are in the chyme, and they stimulate secretion of pancreatic juice, which, as we learned earlier, is high or rich in digestive enzymes. It also causes the contraction of the gallbladder, which is going to release bile into the hepatopancreatic ampulla and ultimately into the duodenum. And finally, it causes relaxation of the sphincter of the hepatopancreatic ampulla, which allows for that pancreatic juice and the bile to be dumped into the duodenum. The other hormone that's secreted by the small intestine is secretin, and that's released by S cells. And it's stimulated to be released when chyme, very acidic chyme, enters the duodenum. And what this does is it causes secretion of pancreatic juice that has a lot of bicarbonate in it. And what that does is it neutralizes the acidic chyme as it comes into the small intestine. So if you recall, the chyme in the stomach has a pH of about 1.5 to 2, but we don't want it to be that acidic in the small intestine. So the idea here is you're going to neutralize that and make it less acidic so it doesn't damage the small intestine. So that's the end of the presentation on Chapter 24. Let's ask you a few questions to see if you recall what we talked about in these last few sections. Starting with this one, you absolutely love plain, unsweetened whipped cream, and you've eaten a cup of it by itself. Which of the following enzymes would be most useful in digesting this snack? The answer is going to be gastric lipase, and the reason for that is that the cream is full of fat, and they indicated that it was unsweetened, so it probably didn't have any sugar in it. The gastrocolic reflex, which one is correct? It initiates mass peristalsis, and this mass peristalsis is what's going to occur starting in the transverse colon, then the descending and sigmoid colon. As you smell food, your mouth begins to water and your stomach rumbles. This signals the beginning of the gastric phase of digestion. Is this true or false? It's actually false. It signals the beginning of the cephalic phase of digestion. The enterogastric reflex, which one is that? I'll give you a second to look and decide. B, it's stimulated by the presence of chyme in the duodenum, and what this does is it prevents or tightens the pyloric sphincter, preventing chyme or slowing down the entry of chyme into the duodenum. Blank stimulates increased secretion of pancreatic juice that is rich in bicarbonate ions. Which one does this? The answer is B, secretin. We've reached the end of Chapter 24 on the digestive system. Be sure to go back and review the Chapter 24 handout that you can print from Converge, and also make sure you go through your reading guide and objectives. There's a lot of other material that I've provided for you on Converge to help you study for the quiz, so be sure to go over that. And there will be an online quiz, and that's scheduled for Wednesday, April 1st. You have 15 minutes to take the quiz once it's opened. You can take it at any time before midnight on Wednesday, April 1st. But once it's open, you only have 15 minutes to take it. I hope you all do well, and we'll meet again for Chapter 25.